Dr. Kathleen Hunt is a physiologist and endocrinologist with a strong interest in applied conservation physiology. Most of Dr. Hunt's research interests center on impacts of environmental and anthropogenic stress on reproduction and health of free-ranging vertebrates, with an emphasis on applications for conservation. She has particular expertise in hormone assay validation and has developed hundreds of hormone assays for alternative sample types that can either be collected non-invasively without disturbing the animal or can be found in museum archives. In this talk, Dr. Hunt will describe how endocrine analyses of cetacean feces, respiratory vapor, baleen, and even teeth have enhanced our understanding of whale biology, including how whales are impacted by environmental change and human activities. Thank you so much, Dr. Hunt.
Well, thanks so much. Should I just jump right in and get going? Everybody
Yes,
logged in? Okay. Thank you all for being here with me. I already see some names I recognize in the audience, so I know some of you already know some of the things I'm going to show you. But nonetheless, let's get going here. So I am interested in stress and reproduction. I really I was trained originally as a sort of a classic behavioral endocrinologist, following small birds around and looking at their behavior and how their individual behavior was affected by hormones. And from that, I've become very interested in really stress physiology and how stress affects reproduction. That's probably my core interest. And I've realized over time that if we can get a good idea at what's going on with individuals, we are then better able to forecast what's going to happen to populations, because population trends really only happen because things are happening to the individuals. So these days, I frame my research in terms of if I can understand individual stress, I can then see how that affects individual reproduction and mortality. Maybe I can get sort of an early warning view of problems that are going to be affecting population numbers in the future. And in addition to the ultimate impact on populations, I'm also just interested in individual welfare. Another reason I study stress is because I actually care about the welfare of the individual animals. I like to study stress and reproduction by means of looking at the hormones involved in stress and reproduction. And I realize I don't have any slides in this PowerPoint about hormones in general. I'm assuming this audience largely knows what a hormone is. But essentially, I do a lot of work with the glucocorticoids from the adrenal gland and the reproductive steroids, so progesterone, estradiol, testosterone, cortisol, corticosterone, those kinds of hormones. And they're all steroid hormones, which all have this classic four-ring structure as shown here. The problem with studying hormones in some of the types of wild animals that I'm trying to study is typically, well, traditionally, we would have to get a blood sample to study these hormones. And when I shifted away from small birds to studying bears, as shown on this graph, and then elephants, which was next, I realized there's quite a few types of animals out there where it's really difficult or even impossible to get blood samples. And also, if you're studying stress, the one problem with blood sampling is that you stress the animal when you're taking a blood sample. So you're changing the thing you're trying to measure. So I spent a lot of my postdoc figuring out other ways we could study stress and reproductive hormones. So then I shifted to whales, and I rapidly realized they may be even more difficult than the bears and the elephants, and possibly the most difficult animals to study. With the whales, we often can't even see them, never mind about getting blood samples. We want to get up close and study their behavior like this, and very often this is the view we actually get. We get these distant, fleeting glimpses. This is a blow of a distant whale here. And so it's hard to even get a look at them or even find them. And blood sampling is virtually impossible. I used to say completely impossible. There's a group working on a blood sampling tag, and that would be lovely if it worked out. But right now there is really no method of safe capture of the large whales. There is this term, the great whales, that I had in the title of the talk. The great whales are all the large baleen whales, such as this southern right whale shown here. Plus the sperm whale, which is actually a toothed whale, but it's also very large. So it's really all the whales that are too large to capture safely, and we just can't get blood samples from these guys. So blood sampling is out. But fortunately, there are sample types other than blood. And the terrestrial wildlife people had been working for some time actually on especially fecal sampling as a possible alternative to blood. The whales really ended up pushing me to consider other sample types as well, because I will show you some data from or some images of fecal sampling from whales, and fecal sampling does work in whales, but even it's hard to even get poop from a whale. So we've we who study whales have been forced to get creative and branch out into other sample types beyond even the poop. So I'll show you a little bit about respiratory vapor. There's teams working on getting hormones out of the little bits of blubber that you can get from biopsy darts, little bits of skin and blubber as shown here. There's hormones in earwax plugs, and then there's baleen, which I'm going to talk about a fair bit today. So all of these other sample types, though, whenever you're dealing with a sample type other than blood, there are a number of things you have to consider before you can have any hope of interpreting your data. And one of the big things is the time frame that that sample represents. So, for example, when we get a blood sample, we consider that usually to be a snapshot of the circulating blood concentration in blood plasma at that moment that you had the needle in the vein and we're actually getting the blood. We think of all the other sample types I just showed you, we think respiratory vapor probably is the most immediate time frame, if I can put it like that, probably representing something like the minute that you were by that whale collecting that respiratory vapor sample. So respiratory vapor does work. I'm not going to spend a long time on it today. This is a slide of a North Atlantic right whale. It's facing away. That's its snout over there and its body here. And this is a picture I took from one of my the research teams that I work with at the New England Aquarium. And as you can see, we just stick a little device on a pole and get it out over the whale, and we can actually get a sample of those respiratory vapor droplets. And yes, there are hormones in there. They are quantifiable. This technique was first tested by Carolyn Hogg, who first was able to detect presence of the hormones. And then I and multiple other colleagues of mine followed along with a couple more papers verifying we are in fact able to quantify the hormones. So there's a difference between just telling that a hormone is there and being able to put a number on it. So we can now put a number on it. The concentration is hard to figure out because you have seawater droplets. These little big drops here are seawater. So there's seawater mixed in, and that gets complicated. But we can measure the hormones, and we are getting some biologically realistic looking signals out of the data. So there are other teams now, especially a colleague of mine, Liz Burgess, who worked with me on this right whale project in the photo and has since continued working with blow. She's got a field project going right now with more blow sampling. And there's other teams now using this successfully on some of the toothed whales like belugas. So blow sampling does work. We can get blow samples pretty, I don't want to say it's easy, but it's easier than getting the poop. It's certainly easier than blood. And they're tiny samples. The hormones are very low in concentration, but I think this is going to work as an alternative sampling method. So respiratory vapor is one option. Then there's fecal sampling. So fecal sampling, this is where the time frame really starts to shift. It's been well established by people working on terrestrial species and getting fecal sampling from the terrestrial mammals that fecal hormones, first off, there are hormones detectable in vertebrate feces, and it tends to represent something like an average of the circulating hormone over the prior 1 to 2 days. And that time frame will vary a little bit. Actually, it can vary quite a bit depending on the gut passage time of that species. And that tends to also correlate with whether it's a carnivore or an herbivore. The herbivores tending to have slower gut passage time. But you've got something like a couple days of hormone represented in the poop. Fecal hormone assays, as I think a lot of you already know, the hormones are getting into the poop by means of the liver. The liver is using the digestive tract as a clearance route, as a mechanism to get hormones out of the blood and sort of decommission them. Hormones don't circulate forever. The signal needs to be stopped at some point. So a hormone is put into the blood, circulates around carrying its message, and eventually there will be some mechanism of breaking down that hormone and removing it from the blood. And in the case of the steroids, they're taken out of circulation by the liver and then either sent to the gut or sometimes the urine, and often split between the gut and the urine. But some of it will come out in the poop. But what comes out is usually no longer the pure parent hormone. It's been altered quite a bit by digestive bacteria and enzymes. Oh, by the way, we have background noise going on. Maybe the organizers can double check and mute other people just so we don't have background noise interfering with things. Let's see if I can figuring out if I could do the mute. I cannot do the muting, so hopefully you guys can mute yourselves. Anyway, back to the fecal hormones. So what we get coming out in the poop is a mix of metabolites derived from the original parent hormone, and that can be difficult to figure out. Sometimes it's not clear which metabolites came from which parent hormone, and there's usually quite a few validations you need to do to be sure that the thing you're measuring actually is derived from the hormone that you're trying to assess. But it works pretty well. And we've gotten this to work for whales quite well. This photo here is actually from a southern right whale, but we first tried this technique in a close relative, the North Atlantic right whale. So I and my New England aquarium colleagues did a number of papers on fecal glucocorticoids and reproductive hormones. We were able to show correlations of the fecal glucocorticoids with exposure to shipping noise. We're able to identify the sex and the reproductive state and the maturity status of the whale just from hormones. Like if we find a floating right whale poop all by itself and we don't know what whale it even came from, we can still correctly identify sex and reproductive status and whether or not the animal was sexually mature, if it was a female, whether it was pregnant, with quite high confidence. So this method works quite well for figuring out what's going on in terms of stress and reproduction with the animal. And there's been a number of more studies now in other species. I actually need to update this list of papers on the bottom because there's been a lot more papers since the last time I revised this slide. So this works well if you can get enough poops to do your study, and that's usually the sticking point is can you find enough poops? I do want to show you one example of a current project I'm working on that does involve fecal sampling, because I think it's a fun illustration of how some of these non-blood sample types can be combined with other new methods of studying animals and synergize really well. I'm actually going to back out of screen sharing for just a second. And if I can, let's see, I'm going to stop sharing so that I can reshare. We have a type of a version of Zoom today that isn't allowing me to share my whole desktop. So I'm going to reshare this separate screen just to show you a little video of how we get poop from gray whales. So in this video, this is a research project I'm involved with on Pacific Coast gray whales off the coast of Oregon with a great team led by Dr. Lee Torres, who works at Oregon State University. And they fly drones over the whales. Drones have been a really exciting new development in whale research. We're able finally to get over the animal and sort of stay with them from pretty close. Instead of being, way high above in an airplane, we can now be like relatively close in a drone and get a really good view of their body contour and their behavior from much closer than you could do in an airplane. So drones have really revolutionized the way we study behavior and body fat assessment. You can look at the contour of the animal and figure out what, is it a fat or a skinny animal? And to some degree, you can sometimes tell if females are pregnant, like are they fat in the back half of the body instead of just in the sort of overall. So one thing we didn't anticipate is that when we're flying over the whale, so here's drone video from a gray whale, there you see it blowing. Look at this brown cloud starting to emerge from the hind end of the whale. We can see from the drone when the whale defecates. And the Oregon State team realized really we can use the drone to do double duty. It can be collecting all the drone footage, but also the drone operator is watching the drone camera in real time, and he can tell us if he sees a poop. And then there's us in our poop boat zipping over to sample the poop while the whale moves on. So we were able to increase our sampling rate quite a bit because the drone operator was able to spot these defecation events for us. I also wanted to show you this video to give you an idea that poops are not always easy to collect. Gray whales turn out to be a species that produces this sort of diffuse cloud of fecal particles that immediately starts to disperse and sink, and you have to race over fast. You can see we are using a dip net back and forth through the water to get as many of those little particles as we can. And we do get enough to assay, but we don't get anything near the original volume of the whole defecation. And you have to do it fast because it's sinking. So some whales make poops that stick together nicely and float. The right whales turned out to be really good for poops. Some species have this diffuse sinking cloud like the gray whales. But even with the gray whales, we were able to get samples and learn quite a bit from the animal. So that's one way we do fecal sampling in the whales, and that's one way we've been able to increase our sampling rate. Now I'm going to reshare it back to my PowerPoint, and we'll go back to the rest of the presentation. So again, that was the Oregon State team, and this still photo is actually another image from one of their drones. So from those fecal samples from the gray whales, as small and dispersed as our samples are, we have been able to get a pretty good measure of fecal glucocorticoids. We have a nice paper actually that we just submitted, headed by this great statistician out of the University of St. Andrews. Enrico Perrotta is his name. I'm sure some of you know him. He has put together this beautiful model demonstrating that we can predict calving probability of a female pretty well if we know her body condition and her fecal cohort of 1 to 2 years before. So basically, the year before she's pregnant, she's making the decision whether to ovulate, essentially, and both her stress levels, if I can call, cord is not just a stress hormone, but I'm going to lapse into calling it that just for simplicity. So just from her stress hormone levels and her body condition, we can predict pretty well whether she's going to make the decision essentially to go ahead and get pregnant the following year. So that's been really interesting to figure out. We've also then been able to tie the cord to exposure to specific other stressors like boat noise and disturbance of various types. That the signal is weak, but it is there. Animals' stress hormone levels fluctuate all the time for many, many different reasons. So one thing about working on stress hormones is there's a lot of stressors, a lot of different things happening to animals, and it is tricky to attribute a certain rise in fecal adrenal glucocorticoids to a certain event that happened to the animal. But sometimes if you get enough data, you can figure out all those factors and add it all up together. And our model's actually working pretty well for both predicting the fecal cort level and then predicting the effect on calving. We've also just recently got another paper out by Dr. Alejandro Fernandez Ajo on fecal testosterone in the males. And we've finally realized, we've had this data for a while now, and Ale just recently went back and looked at our historic data. We realized that the large males are showing a rise in testosterone as breeding season approaches, but the small males are not. So it's something that's happening just really in the adult males. And we just hadn't realized that we were sitting on that data. So it was a really interesting example of being able to pull out information on reproduction in the males as well. Poop works for whales is the message I want to leave you with. I want to shift now and look at a totally different kind of sample type. So I spent about 10 years following right whales around trying to get poop and respiratory vapor blow. So poop and blow studies were my bread and butter for a while there. And they were working, but I was always a little frustrated by the fact that we couldn't get repeated data over time from the same individual. I really wanted to assess one individual story over time. But as is common in wildlife biology, you see an animal once, you get one sample, you may not see that individual again for years. And it's quite hard to really follow an individual over time. And then I became aware that whales have this other sample type, baleen. Let me get you oriented here to any of you guys who don't know what a snout of a right whale looks like. This is the North Atlantic right whale. This photo, by the way, was taken by Liz Burgess at the New England Aquarium. The whale has its mouth open. We're looking basically at the top of its upper jaw. It's moving to the right. This is the tip of its snout, if you want to think of it that way. This is its upper jaw here, or what we call the rostrum. And what is all this stuff here? This is baleen hanging down from the upper jaw. The lower jaw is underwater. This whale is swimming through the water with its mouth open, just kind of scooping up food in its path. So what is this stuff here that's hanging from the upper jaw? Let me back out a little bit more. And before I move on, let me just say these are strips of keratin. This is a keratin tissue. I'll come back to the baleen and I'll get you oriented to how these little strips here are all lined up and how they grow. But let me first give you a bigger picture. A lot of vertebrates tend to surround their bodies with an outer layer of a keratin tissue. So we have keratin in the epidermis of our skin, and most vertebrates do something similar. Keratin is a sturdy waterproofing protein. And most of the vertebrates have basically got a lot of keratin in the epidermis of their skin. It's how we keep from drying out, we terrestrial mammals. And for marine mammals, it's how they keep from losing body water to the salt water of the ocean. And so we've got it in our skin. And a lot of vertebrates grow various things from the skin that are also made of keratin. So fur is made of keratin. This is a tail of an elephant, elephant tail hair here on the left. And then just going left to right, we've got human fingernails, seal whiskers. These are elephant toenails, which I have been studying. This is a spiny echidna, a short-beaked echidna. And that's got the little spines are made of keratin. Rhinoceros horn and artiodactyl horn are made of keratin. Here we have some reptiles. This is a tegu lizard. Lauren Buck and Danielle Dillon and other colleagues of mine and Lucas Zena have been working on skin from tegu lizards. They shed patches of their skin. Here's a snake skin, turtle claw, bird feathers. This is also from Lauren Buck's team. So all of these are keratin structures, and you can see there's a variety of sort of funny shaped things that grow out of animals' bodies that are made of keratin. So keratin accumulates hormones as it's growing. Whatever hormones are circulating past in the blood, past the growth zone of the keratin tissue, some of those hormones just seem to seep into the growth zone and end up being deposited into the keratin tissue as it's growing. Every tissue I've shown on here has publications out on that particular tissue demonstrating that there are hormones in that particular keratin tissue, typically all the steroid hormones and thyroid hormones too, and other things also, and besides. But I've been focusing on steroid and thyroid hormones, so I'll keep my focus on those. So some of these tissues are kind of long and they're growing steadily and continuously from a base region and sort of extending outwards as they grow, like our hair and our fingernails. They're growing from a base and getting longer and longer. Any of you guys that have long hair that are listening to me, consider one long piece of hair from your head. That hair is a time series. It's grown over several months. And it's got hormones that have been laid down inside of it. And I could get a hair off of your head and look at the tip and see how stressed you were several years or months ago, and look at the base and see how stressed you were last week, and essentially get a time series of your hormones over time. I'm saying that crudely, like boiling it down to how stressed you are is a crude way of describing what stress hormones really mean or what the adrenal hormones are really doing. But I'll just use that simple shorthand to get the point across. So baleen is kind of like hair and fingernails. It's long and skinny and growing from a base region. So let me get you a little more oriented here. This is the mouth of a species called a Brutus whale, and this animal is lunge feeding. So some whales do this thing where they leap up with their mouths open and they kind of scoop up a bunch of fish. So that's what this whale happens to be doing. What it's going to do is close its mouth, and this white stuff here on the right side and the left side of its mouth, that's the baleen. Here's a close-up look, and I think you can see the baleen. It looks almost furry here. What's really going on is it's a whole bunch of these pieces all lined up parallel to each other. And each piece is called a plate. And they're shaped kind of like an isosceles triangle. They're usually wider at the base and skinny at the tip. And they're anchored at the base, anchored in the lower jaw, and that's where they're growing. So that's the growth zone. And as it's growing slowly over time, it's extending downwards, and the tip is just free hanging in the mouth. The tip gets eroded away over years as the whale is feeding and just swimming through water. So the tip, the baleen is getting skinnier as you go through the tip because it's eroding away. And eventually, in an adult whale, there's a steady state where it's eroding at the tip just as fast as it's growing from the base. So we've got here a time series. The base of the baleen was grown, well, I should say one important thing about baleen that is a little unfortunate. We can't get baleen out of living whales. We have to wait for a whale to die and wash up on shore. But unfortunately, that actually happens pretty often. So we do get a lot of baleen from these strandings, from necropsies of stranded specimens of whales that died for some reason or other and ended up on shore. So when we get a piece of baleen out of the mouth of one of these stranded specimens, my assumption is that the base of the baleen was grown just before the whale died. And then regions of the baleen further toward the tip were grown farther back in time. And the tip was usually grown several years ago. And you've got a time series here along the length of this piece of baleen. And you can drill little bits of baleen powder all the way along the baleen plate and assemble a story of that whale over time. So what is the timeline, though? And here I want to make a general point that with any of these non-blood tissues, you need to figure out the timeline that sample represents, which may involve, for the keratin tissues, figuring out the growth rate of that tissue, and also when in the year was that tissue grown. There's some tissues like bird feathers that are just grown in 1 month of the year. For example, in the Northern Hemisphere, birds typically grow their new feathers in August, at the end of the breeding season. And then they just keep those feathers for a whole nother year before they replace them. So you want to figure out, okay, when did this sample originally grow? And if it's a continuously growing tissue, okay, what's the growth rate of the tissue? So how do we figure that out? In the case of the whales, we have been able to lean on the fact that most of the great whales have annual migrations. And especially the baleen whales tend to have these very consistent migrations where they're going back and forth. In summer, they go toward the poles, toward colder waters, either toward the North Pole or toward the South Pole, where they feed. It's a summer feeding season. And then they turn around in the fall and go back basically toward the equator, toward warmer waters, where they typically will have their calving and their breeding grounds. I should mention a lot of the great whales fast to some degree, or at least reduce the amount that they're eating when they're in their calving grounds and mating grounds. But we now know that actually a lot of them do keep eating at least somewhat, and some of them, in fact, keep eating quite a bit. So they're eating typically different prey in their summer grounds versus their winter grounds. And there are these things called stable isotopes. So you all have heard of, the common elements, nitrogen and carbon. And cast your mind back for any of you for whom it's been a while since you heard about isotopes. Isotopes are just different forms of an element that have different numbers of neutrons and have different weights. And sometimes there's an extra neutron. For example, most carbon has 6 protons, 6 neutrons, so atomic weight of 12. Every now and then you find a carbon atom that's got an extra neutron and it's got an atomic weight of 13. So there's this rare isotope, carbon-13, and it's a stable isotope, meaning it's not one of the radioactive ones that'll fall apart. So that's what it means to say something is a stable isotope. So turns out the rare stable isotope, carbon-13, has a characteristic ratio with carbon-12. And that ratio is different in different places on the planet and in different prey species. So essentially, we can take a piece of baleen. Here's some data from a bowhead whale. This is different locations along the length of one big long baleen plate from a bowhead, different centimeters from the base. And we can measure the ratio of the rare isotope to the common isotope. And you'll see that ratio is changing as you go along the baleen, and it's cycling. It's going up and down and up and down and up and down. And basically, as you're going along the baleen, what you're seeing here is the whale is eating summer prey and then winter prey, and then summer prey and then winter prey, summer, winter, summer, winter, summer, winter. And in many species of whales, not all, but many of them, you will get this characteristic annual cycle in these stable isotope ratios. So this has been a godsend for me. There is a pretty robust literature already about analysis of stable isotope ratios. And the people who do these analyses are typically actually interested in, okay, what are the whales eating and where are the whales? And really often looking at trophic ecology questions. But I'm interested in this data. Well, I have started to get interested in trophic ecology as I do more stable isotope analyses. But the reason I first started getting interested in stable isotopes is this is telling me the timeline. I can just count the number of peaks, and that's the number of years. And I can even assign each little point on the baleen an estimated season. Like, okay, this area here was summer 2013, and this next little area was fall of 2013, and so on and so on. So this is where we get our timeline. And now we can go to the hormones and measure hormones at all these places along the baleen plate. And now we know we have an estimated season and month associated with each hormone data point. So that is what gives us the timeline. We've had to work out that timeline for every species. Baleen comes in different lengths. Different species have different lengths. For example, bowheads have really long baleen, and often the baleen captures a 15 to 20-year time span. So that's not the whole lifespan of the whale. That's going to be the most recent, the last 15 to 20 years of that whale's life. That's for an adult. Young whales always have faster growing baleen, so you'll get fewer years represented in baleen from a young whale. But in an adult, you'll get 15 to 20 years. Here's a southern right whale. So in right whales, we get about 10 years. And I'm about to shift over to showing you some data from right whales. So again, let me again just get you oriented to how the baleen is positioned in the mouth. So this is a skeleton of a southern right whale with the baleen arranged as roughly as it would be in life. So attached at the upper jaw and hanging down in the mouth. So you can see all these little separate plates are all arranged in essentially 2 big curtains, one on the right side of the mouth and one on the left side of the mouth. Now, each piece of baleen, or again, those are called a plate. Each plate of baleen is roughly the shape of an isosceles triangle. So I've shown you this blue overlay of basically one piece of baleen. Keep your eye on that isosceles triangle. I'm going to use that triangle to get you oriented for the graphs I'm about to show you. So on these triangles, one triangle is roughly the shape of a piece of baleen, and the wider end of the triangle is the base. That's the growth zone. That's the newest baleen. And the tip is the oldest baleen. Okay. So I can, as I mentioned before, we can get the timeline from the stable isotopes. We can measure hormones. We'll get a bunch of hormone data. We'll see the hormones going up and down and, having various patterns. Does it really tell the truth? That's my quick shorthand for, is that hormone data really corresponding in any realistic way with the actual physiological history of the animal? So again, a lot of you guys, I'm sure, know this already, but when you start with a new strange sample type in a new species, you need to do a lot of validations. I divide validations mentally into 2 bins. First, I tackle what I call the assay validations, which are roughly, can we measure the thing correctly? Can we measure the concentration and put a correct number on that concentration? So that includes the validations like parallelism validations, limit of detection, your extraction efficiency, accuracy validations. So suppose you do all those validations and you conclude, okay, yes, I'm measuring it correctly. If I measure this sample and it comes out with 3.2 nanograms of progesterone per mil of extract, that number's correct or reasonably correct. You still don't know whether that number really corresponds to anything meaningful about your animal. You need to do a whole other type of validations too, which are called, or I call them, biological validations. For biological validations, you want to compare your data to animals where you know something already about their physiological state. Classically, we used to do this by injecting various things into the animals and basically either forcing their hormones to go up by giving them a shot of the pituitary hormone that will make the adrenal or the thyroid gland or whatever release the hormone you're interested in. Or sometimes injecting animals with a radio-labeled version of the hormone and then seeing where the radioactivity comes out in the sample. We cannot do any of these in whales. We can't do any of these classic pituitary hormone challenges, they're called, or the radio label infusions. What we can do is look at individuals of known history. So in whales, we just have to accept that we cannot do a lot of the classic validations, which is always a sort of a nagging problem. But it turns out in some populations, you can identify individual whales. And in some research teams have been doing a wonderful job following individuals over time and assembling these sightings records of when a given female was seen with a calf, when a certain individual was entangled in fishing gear. And if you're lucky, you may be able to get a sample of an individual where you know something already about their reproductive history. So what I'm going to do now is show you my first case, and it's still the clearest case, the clearest example of how biological validations work. And it's the case of this right whale, Stumpy. So this is a North Atlantic right whale, which is a different species than the southern right whale that I was just mentioning. The North Atlantic right is its own species, and it is critically endangered. It's been, unfortunately, in the news quite a lot recently because right whales have not been having a good decade. They've been in a pretty worrying decline recently. So I used to study right whales quite a lot with the New England Aquarium. And there was this is one of many sad cases where individuals have been found dead. This particular individual died of a ship strike in 2004. She was a known female. She had a name. We name all the right whales. She had a number. Eubelina glacialis 1004 is her number. Her name is Stumpy. So she turned out to have a pretty good sightings record. We had, again, we can tell right whales apart from each other. They all have different patches on their heads and you can identify them. And she had a pretty decent sightings history. And this was long before I started working on whale hormones. And but I am forever grateful to Michael Moore, who is a veterinarian at Woods Hole, who's the fellow here in the red jacket. He attended a lot of large whale necropsies in his position with Woods Hole. And to his great credit, he had the foresight to collect samples even in cases where we weren't even sure if the sample was going to be useful. And he, for example, collected baleen from Stumpy and from many other whales as well, just in case it might be useful someday. And in the end, when I realized that baleen contains hormones, which we did not know till relatively recently, it turned out Michael Moore had carefully kept and saved these baleen samples from Stumpy. And I'll just mention, it's not easy to get these big baleen pieces out of the mouth of a dead whale and, carry them to a vehicle and store them somewhere and get them cleaned of the soft tissue. It is not a trivial task. So I'm very grateful to Michael Moore and all the necropsy stranding teams who take the effort to collect these samples. So what do we know about Stumpy? So here I have that isosceles triangle again. So imagine we have one baleen plate from Stumpy. This is going to be the baleen plate that Michael Moore gave to me from Stumpy. So I'm going to graph for data as if the baleen plate is oriented like this with the tip over here on the left. That's the oldest baleen grown, we think, 9 to 10 years ago. And then I'm going to show you data going along the plate from left to right. And we're going forward in time as we go from left to right with the base of the plate way over on the right. And that's going to be the newest baleen. So for Stumpy, we actually knew her specific baleen growth rate because a friend and colleague of mine, Nadine Lysiak, had in fact just finished up a PhD thesis in which she had been measuring baleen growth rate of North Atlantic right whales, including of Stumpy. So we knew her baleen growth rate. And we had a pretty good estimate of what the timeline would be. So Using her known baleen growth rate, I basically took the centimeter measurements of each point along her plate and converted it to an estimated month and year when that little bit of baleen was probably grown. So I have the estimated growth now. And that came from Nadine's data and really from her stable isotope data. That's the stable isotopes again that gave us that. So then I went to her sightings record. So in Stumpy's case, we had 2 times in the past 9.5 years when we knew she must have been pregnant. She had a young baby at the very end of 1996. So we knew she must have been pregnant the year before that. And when she was killed, she was actually on her way to the calving grounds and she had a full-term fetus inside of her. So tragic loss for the population. That does mean that we know, therefore, that she was pregnant during 2003, the year before she was killed. So we've got 2 definite pregnancies. And I have highlighted in yellow the predicted, what I thought were her pregnancy periods. And that's based on the date of birth of the calf or the date that she was killed with the full-term fetus. And then I've extended the pregnancy backwards 12 to 13 months beforehand. These yellow bars actually bracket 13 months based on what we thought at the time was probably the gestation length of right whales, though you'll see we may have been underestimating that gestation length. So 2 definite pregnancies, and then I went and measured progesterone. So I basically drilled a little bit of powder every 4 centimeters along the entire length of this long baleen plate. And using methods I'll give you a snapshot of pretty soon. I used a pretty simple methanol method to get the steroids out of the baleen powder from each of these little spots along the plate. And then I just measured progesterone in the resulting extracts. So here's the data. Look at the y-axis here. This is a log scale. So you can see she has 2 regions of extraordinarily elevated progesterone. So this progesterone here is about 100 times higher than her baseline progesterone down here. And again, in her second pregnancy, strongly elevated progesterone. Progesterone typically goes up in pregnant female mammals. So this was an excellent match of her known calving history to the endocrine data. By the way, I had been wondering if we might have missed a pregnancy of hers somewhere around 1998, 1999, because you notice we didn't see her for a while. And we expect a healthy right whale to produce a calf about every 3 years, and she went 7 years between these 2 calves. But just from her endocrine data, it's looking strongly likely that she did not get pregnant in here. And in fact, looking back at the records from the whole population, it turns out a lot of females in this time period switched to very long intervals between calves. There seems to have been a disease going through the population in the late 1990s. So that lines up with the reduction in calving in the whole population that happened in that time. So this was the first time I'd assessed a hormone along the whole length of a baleen plate of an individual of known history. And it was really beautiful. I was really, really pleased with this. And this was really my first hint that this method was really going to tell us something useful about the animals. By the way, look at when her progesterone goes up. It goes up quite a few months before we thought it would. And that was actually, looking back, I now realize that was our first hint that gestation length in the right whales and also the bowhead, they're all closely related, is probably longer than we had originally thought. They're probably getting pregnant earlier than we thought and having like something like maybe a 15 to 16-month gestation. I don't want to be super firm about that because we're still not certain what the temporal resolution is of a single point on the baleen. Is that like a week, or is it a running average of the surrounding 6 months? So that's what I call the temporal resolution. I was not sure of the temporal resolution, but look how fast her progesterone drops, aligning with the appearance of this newborn calf. That gives us a clue that there may be some blur in that temporal resolution, but it's not enormous. It's not enough to account for this big gap here between her first rise in progesterone and the 13 months before birth window that I had expected. So we're thinking, long story short, we're thinking their gestation length is probably a little longer than we originally thought. So I'm just going to show you that one case of doing a biological validation, but I will give you an array of papers here in the bottom just to sort of give an idea how this field has unfolded. So myself and my colleagues at the New England Aquarium, and also Rafaela Stimmelmeyer from the North Slope Borough, we put out a bunch of validation papers demonstrating this really beautiful alignment with progesterone data, matching calving history in a couple of females. We got pretty good matches of glucocorticoids to known entanglement events, which is sort of our go-to definite stressor. Like a whale entangled in fishing gear is definitely a stressed animal. And then I jumped ahead and decided I'm going to do all the assay validations all at once. I published a paper that had No, it was a paper consisting of 64 parallelism tests. It was, I just thought, I'm just going to test 8 different hormones in baleen of 8 different whale species and just see if this method is going to work generally for the whales. And it did. All the hormones were there and all the baleen of all the species. So since then, the method has been, we've done a bunch more validations, and the method has been applied successfully to ask and answer welfare questions about whales. There was this famous bizarre situation where southern right whale calves were washing up dead, covered in little wounds on their backs from gulls biting their backs. And we were able to show that the number of wounds corresponded with the glucocorticoid patterns in the baleen. The more wounds, the higher their cort was, basically, to the point that it looked like these gulls were actually killing the whale calves. Bizarre episode that's actually still going on to some extent in this one region in Patagonia, at this one calving ground. And the method works in other species too. There have been papers on humpback whales. It works for blue whales. It works for fin whales. Nadine has published a beautiful paper showing that bowhead whales have this extended gestation length too, and a lot of other interesting features. We have Alejandro Fernandez-Ajo, who I mentioned had the fecal testo paper on the gray whales. He also now has a baleen paper out on gray whales showing that so there's been a die-off in gray whale juveniles in recent years, and Alejandro was able to look at the glucocorticoids again, and he could see the cort elevating in these gray whales. He was able to get an estimate of whether the thing that killed them was brief or long-lasting. Like, did their cort go up just the month before they died, or did it go up a year before they died? It turns out the cort starts to go up about 8 years before the whale dies and continues to go up until they die. So whatever is happening to those subadult gray whales, it's starting about, did I say years? I meant months, 8 months. It's starting more than 6 months before the actual point of death, which was a useful thing to know. That gives us an idea, okay, where are the whales 8 months before they die? What's going on in that feeding area? So sometimes we can't tell why the cort goes up, but we can tell when it goes up. And in the case of a migrating species, that tells you something about where the population is happening, where and in what season. And that alone can be really useful data to have. So I think of this really as baleen tells the truth. There's still some uncertainty about this time frame. Like I said, the temporal resolution is probably the question I most want to answer is a single point on the baleen an average of the surrounding 2 weeks, the surrounding 2 months? Like, how precise is that data point in terms of its timing? But generally, the method is working really beautifully. To the point, actually, before I go to the methods, let me just back up and say, the moment I saw this graph, the whales rocketed in my mind from one of the, if I can put it this way, one of the worst species to study in terms of how difficult it is to get data from them, suddenly to one of the best. Because all of a sudden they went from the whale where I couldn't get a hormone sample at all to the animal where I couldn't get a hormone sample at all, and all of a sudden became the animal where I could get 100 repeated samples in a row across an entire decade. And that kind of data set is rare for any species, never mind about whales being difficult study species. We don't get this kind of data set for terrestrial animals either, or even for captive animals usually. So this is an extraordinary time series to be able to get, especially from the large whales. And it's been really exciting. That said, like I said, we can't get baleen from living whales. So we typically have to wait for a whale to die or have to work with the samples that are available. I have had a number of cases where I design a beautiful project in my head and then go to the museum that I think will have those samples, and they say, well, what we have is all these gray whale plates from the 1930s. Do you want those? And so you have to sort of design your project around the samples that are available. But nonetheless, we've been able to sort of go to different museums and piece things together, and generally have been successful at addressing the question that we most want to address. One thing that I've really been excited about the length of these time series, is I've finally been able to get a peek at the long-term effects of stress. An animal gets stressed in 1 year. We often, if we can study it at all, we study it that year, maybe the next year. What about the year after that, and the year after that, and the year after that? We know that stress can have multi-year effects. An animal can be stressed 1 year, and 5 years later, it still might not be back to breeding normally. And these long-term data sets give us the ability to see what happens in multiple years after a stressful event. And sure enough, there are actually long-term impacts that last quite a bit longer than 1 year. So that's generally the overall idea. Let me run you through the methods pretty quickly. We've been using sort of embarrassingly simple methods, as I think of it, on the baleen. We simply use a handheld drill to drill a little powder from a given sampling site. There's the powder being collected on a little wave paper there. This, by the way, is Ali Case working on a piece of blue whale baleen from the Smithsonian. And we stir the powder, we weigh the powder. We've managed to minimize the amount of powder that we need because these are quite rare samples and we don't want to just drill them to oblivion. We want to just make a little tiny groove. And we've gotten it down to 20 milligrams, and maybe, I think we're pushing it down to 10 and possibly getting it down to 5. So we're minimizing how much we need. It turns out that once you've powdered a keratin sample, methanol, you can usually just mix the powder with methanol, and most of the steroid and thyroid hormones will just move right into the methanol. We're not getting all the hormones out that way, but we're getting a consistent enough amount that we can see biologically plausible patterns. And then we've been using immunoassays. I have been collaborating now and then with a colleague who I think is in the session here today on using mass spec on some of these samples as well to really spot check, are the hormones really the hormones that we think they are? But our go-to method has been the assays, partly because the immunoassays are still overall still seem to be better when you're dealing with very tiny samples with very low concentrations of hormone, and you only have like a few microliters of your sample. The immunoassays can still function at very, very tiny sample masses and very tiny concentrations, and that's been one really helpful thing about the immunoassays. So that's the story in a nutshell. Let me give you a quick peek at a few more bits of data from other sexes and other species. So we've been able to get a look at testosterone cycles in males. I'm just going to show you 3 slides in rapid succession, just showing you for this bowhead male. Here's his lovely annual stable isotope cycles, so 13 years. Here's his testosterone, and look, it's going up once a year. So males, turns out, have these lovely, predictable annual testosterone peaks, which has helped us clarify the season. And again, these are migratory animals, so that helps us clarify the location of the mating grounds, which has been really useful to figure out. We can get a hint at sexual maturity. Here's a baleen plate from a young male. He was actually the smallest male in our study. And you can see his testosterone here doing not much. And then he starts doing little annual testosterone cycles. And I think he was just starting to make a bigger testosterone cycle right when he died. So we've been able to see the onset of sexual maturity, which turned out to be at a much younger age than we thought in bowheads. I'm not going to say this male was necessarily making functional sperm or actually mating yet, but he was having annual testosterone cycles at least. In older individuals, we sometimes see testosterone peaks getting smaller and faltering a little bit, and cyclicity wavering a little bit. Basically clues that some of these species might be experiencing reproductive senescence, so failure of reproductive cycling in some of the very oldest individuals. This is a graph of testosterone over time from an individual who's probably over 150 years old. So bowheads live quite a long time, in case you guys didn't know that about bowheads. We can look back in time. So one great thing about keratin is it turns out that the steroid and thyroid hormones, which generally are very robust and stable molecules anyway, you put them in keratin, which is a very dry, sturdy protein substrate that seems to be holding them. And the hormones last a long, long time. This is a data set showing, I'll just draw your attention to the pink line at the bottom, which is progesterone. This is hormones over time from the 1950s up to 1964 from a big old female bowhead. And we've just got the isotope data from her. It looks like she had 5 to 6 years between calves. So we suddenly are able to measure calving cycling and basically reproductive rate in populations back in the past, more than 50 years ago. We've got another project going on in going back to World War II, 1930s and 1940s, and we're getting beautiful progesterone pregnancy patterns, testosterone peaks in males, and really interesting changes in the adrenal stress hormones that seem to correspond with the resumption of commercial whaling when World War II ended. That's using this beautiful Smithsonian archive. And not to steal the thunder too much from the talks we're going to give in a couple weeks at the Society for Marine Mammalogy meeting, but we've also got data now back into the mid-1800s and beautiful pregnancy cycles. So I've been startled and very happy with how resistant to degradation these hormones seem to be when they're in a keratin tissue like this. I don't know how much further back in time we're going to push this, but we may be able to go back to museum specimens from, a couple centuries ago and reconstruct some information about stress and reproduction back in eras when human impacts on animals were, I won't say were absent, but were different than they were today. For example, back in the 1800s, We did not have global climate change really happening yet. We did have commercial whaling though. So, there were human impacts, but they were different human impacts. So I want to wrap up by just going back to non-whales. This approach generally will work on any keratin tissue. I have students working on elephant toenails and elephant tail hairs. We're getting nice, lovely time series out of those sample types. And I'll go back to my big slide I showed you at the beginning about all the keratin tissues. I didn't choose these particular animals randomly. All the sample types shown in this graph have papers published showing that we can reconstruct hormone patterns over time using the various sample types from the animals in these photos. And I will actually draw your attention to those ones that are growing, that are sort of long and skinny and are growing steadily from a base region. Those will give you a continuous time series sort of data set. Some of these other sample types will give you more a snapshot of that tissue grew last August, and here's what was happening last August to that animal. So for example, that's how we interpret bird feather hormones typically. So you have to, again, think about how is that sample growing. In summary, I'm rather grateful to the whales. They've forced me to get more creative and look at more types of samples. I probably would never have considered blubber or earwax or blow or baleen, any keratin tissue, if I hadn't been working on whales. And really the whales have pushed us to get more creative. And I will wrap up by saying, it's not just baleen and keratin tissues. There's a lot of papers that have been coming out on additional sample types that I didn't have time to go into today, but I just want to mention we've got hormone papers out now on teeth and tusk, on bone, hoof, egg yolk, skin, fish opercula. I often say to people, I can't look at animals anymore. I was on safari a couple of years ago in South Africa, and I could not stop looking. A giraffe would walk past. Everyone else is taking pictures of the giraffe's face, and I'm staring at the giraffe's butt, going, "Look at those tail hairs." So once you become aware of how many different tissue types do have endocrine information, you sort of can't really look at animals the same way again. I now suspect that the steroid and thyroid hormones permeate the entire vertebrate body, that any tissue type anywhere in the body, even teeth was sort of the really, if we could get hormones in teeth, I think we're going to get hormones in every single tissue. The hormones are low in teeth and tusk, but they are there and they are detectable. So again, you can get creative. You do want to bear in mind the various issues and puzzles that you need to work out. I like to remind people, what is your research question? Just because you know how to use a hammer doesn't mean everything is a nail. Like just because tail hairs have hormones doesn't mean, that tail hairs are going to be the thing that will help you answer your research question. So think about what is your research question? Will these sample types be useful for you for answering that question? If so, you do have to figure out your timeline. You do have to do your validations. I will warn you, a lot of these samples, especially the keratin samples and the teeth, turn out to have low hormone content, and sometimes they're tiny, like especially if you're trying for annual growth rings in small teeth. That's my worst case scenario right now. You get down to these questions of what's your minimum sample mass, what's your limit of detection. Again, I've ended up leaning on the immunoassays actually more and more, really, rather than mass spec. Because it turns out I can push the limit of detection in the minimum sample mass a little farther with the immunoassays. But don't give up. If you have tiny samples and tiny hormones, if your samples are falling off the edge of your standard curve, don't give up. Email me. I can help you figure out what to do. And usually we can find some way to pull some useful information out of even those samples. So I will close it up there. I have a list of funders at the top that I want to thank, and I really want to especially thank the Epley Foundation, which was the first funder that was willing to take a risk on the keratin tissues and especially on the whale baleen. And if it hadn't been for that core funding, we never would have been able to start all the bigger projects later that then later got the bigger funding. So, and I have to thank all my colleagues on all the previous projects I mentioned and all of my friends around the world working on similar tissues. It's really it's a team effort involving a whole field of scientists, not just one institution. So I'll close it out there, and I will thank you very much. And if anybody has any questions, I'd be happy to address them. I guess I will stop sharing at this point as well.
Thank you
Thanks,
so much, Dr. Hunt, for that wonderful discussion. If anyone does have any questions, we have a couple of minutes for questions. You're welcome to put them in the chat, please, and we can go through them one by one. As we get some of those questions out of the chat, Dr. Hunt, if you don't mind, I'll start with the first.
Sure.
I'm wondering, as you were showing the, the blow samples and the feces and then moving to the baleen and some of the other samples, were you doing any kind of correlation studies to show that you knew an animal was particularly stressed at a certain time from their blow, and then you were able to kind of find that moment in the baleen or any of the other samples you discussed, maybe the elephant or any of the other animals?
I often get the question of have you compared to blood and have you compared across sample types? They have such different time frames that it's turned out to be quite difficult to compare. For example, occasionally we do get a poop and a blow sample from the same whale. That's been very rare. There's only been in fact, I can only think of one case where we actually succeeded at that particular task. And poop and blow, we actually did have a reasonable correlation. We had high progesterone in the blow, and the whale did turn out to be pregnant in her poop, and she in fact turned up with a calf the next year. So we actually had the sightings record to back it up too. So there's been a few cases like that where we could at least verify the state of the animal. I find it to be not useful to try to do direct comparisons of the absolute values of the hormones across sample types, because the range of the hormone is going to be different in each sample type. But also just that time frame is so different. But that nonetheless, there are some cases where we can do the comparison, especially in bowhead whales. So bowheads are the whale where we're usually most able to get a bunch of different sample types from the same whale, and that's because there is an indigenous legal subsistence hunt for bowhead whales, both in Arctic Canada and I think also in Greenland. And in Northern Alaska, and I've been working with the Northern Alaska team. So for example, from some of those bowhead whales that are pulled up on shore by the whaling community of Native Alaskans, we've been able to get poop and urine and blubber. We can't get blow anymore, but we can get blood and do some comparisons that way. And generally, again, you have to still bear in mind that difference in time frame that each of those samples represents, but they will tell the same story about the general state of that animal, like is this female pregnant or not? Is this generally a stressed whale or not? So rarely we've been able to do that, and they do correspond. Usually though, we can't do that. Usually we just can't get the same sample types from the same whale because it's so hard to get these sample types. So often I end up leaning on the type of validation I described in the talk, where instead of comparing to another sample type, I compare to the sightings records, to like, what do we know about that animal just from the sightings record? When did she have a baby? Can we compare to sightings? And that's usually what I lean on the most for validations.
Wonderful. I do have one more question for you, if we still have one moment. As we were looking at the baleen and you were describing kind of going along the time course, this is maybe a naive question, but it made me think of tree rings, right? Where you have the deeper parts in the warm months and then the more dense areas. Is there any visual cues in the baleen that tells you if they're feeding or not, and therefore the plate is growing more?
Thought there would be. So baleen sometimes have, you know how sometimes there's a little ridge on your fingernail, or sometimes a little white spot, sometimes a little horizontal line. Baleen sometimes, actually fairly often, will have these little kind of horizontal unevennesses in the surface. Like if you run your hand along the flat part of the baleen, there's little bumps that you're going over, little tiny bumps. And originally I had in mind those might be something like tree rings, like maybe the baleen is thicker in spring every year or something like that. And I ended up concluding that it was not helpful at all. So at least in the baleen, I still don't know what those ridges are or why they're there, but they did not seem to correspond to years. They didn't line up with the stable isotope cycles, put it that way. But I haven't given up. And I will also mention, though, that the teeth do have annual rings that look much like tree rings, in fact, with a light area and a dark area, and then a light area and a dark area. So the teeth we are approaching exactly like tree rings.
That's wonderful. Hannah, that's all the questions I have. If you have anything else in conclusion?
No.
Sure, one more question.
I think that,
Yep.
yes.
I just answered this one more question quickly in the chat. Yes, we can measure mercury in the baleen. I've got a grad student now I'm working with who has managed to minimize some of the analyses for toxicants require a pretty large sample volume, and this grad student that I'm working with at George Mason University has managed to minimize the volume needed to do some of the toxicant analyses. So we think we're going to be able to add PFAS analyses and specifically to our baleen work, which is really exciting. And yes, I really would like to look at heavy metals and traditional contaminants and emerging contaminants and see those potential impacts on reproduction as well. So great question. So thank you all. And if I need to sum up, I know we're at time now. I'll just say I know a lot of you out there are scientists. a lot of veterinary students, a lot of wildlife biology students, and just be creative. And if you have a question and you can't think of the right sample type that might address your question, always just feel to reach out to me. You can find my email address if you just Google my name and add whales. You'll find my papers and you'll find my email address in those papers. Feel free to reach out. And there's a whole community of people who are eager to help you. We all talk to each other, and we're all a very collaborative, helpful community.
Yeah. Thank you. We do have
Thank you
one
so
quick
much.
question. Have you
One-
looked at
yes.
T3 in blubber at all?
I
Dr.
have
Hunt?
not. I've not been running blubber in my lab because I was so busy with the Baleen. I basically Nick Keller's lab was developing the blubber assays, and I was like, "You go for it, Nick. You do blubber. Steve Tremble, you do earwax. I'll do Baleen." We were all sort of swamped with all the validations. So I have not yet run blubber in my lab, but it's a very well proven technique now. And yes, you can measure thyroid hormones, not just T3, but you can get a variety of thyroid hormones in blubber, T4 and reverse T3, other variations on the thyroid hormones. Typically, all these samples will have the thyroid hormones as well as the steroid hormones. Thyroid hormones are also small, robust molecules. They're also somewhat lipophilic. They're able to cross epithelial layers and like get into keratin tissues. So anywhere you find steroid hormones, you typically will find the thyroid hormones as well.
Wonderful.
All right, thanks everybody.